Hi, grade 9. Are you fine? Today, we are going to study the in vitro digestion of starch. This is the chapter 1, activity 2 for grade 9. The objectives to distinguish between in vivo and in vitro digestion, mechanical and chemical transformation, to define enzyme and hydrolysis, and finally, to conduct an experiment that transforms starch into maltose. First, let we see the difference between in vitro digestion or in vivo. For example, the digestion that occurs inside the body in normal conditions, it is called digestion in vivo, inside the organism, inside the living organism. Okay, in vivo digestion that occur inside the body based to body conditions such as temperature and time. Uh, for example, if we eat some food, it will be digested inside the digestive tract under the conditions of body. While in vitro digestion occurs outside the body in laboratory, for example, based on artificial conditions that are identical to those inside the body, that are identical to the temperature of the body. For example, we conduct experiment at 37 degree because it is the body temperature. Now, mechanical digestion occurs under the action of digestive muscles. For example, in the mouth, there is mastication of food. In the stomach, there is churning. In the intestine, there is churning and stirring. The chemical digestion of food is accomplished by the enzyme. Okay, a chemical substance secreted. The enzyme is a chemical substance secreted by the body and ensures the simplification of complex molecules. The complex molecules exist in the substance that we eat, which is called substrate. Under the action of enzyme, there will be molecular simplification, and the result is the product of digestion, which would be the nutrient. Now, an in vitro digestion of starch, starch-based. We take a sample. Okay, at first we take two tubes, A and B, and we put in the first starch with the saliva, and in the second starch with water. Okay, in tube A there is starch and saliva. In tube B there is starch and water. And we maintain the temperature at 37 degrees for 20 minutes in order to ensure the digestion of starch in vitro. Okay. At the beginning and at the end of the experiment, we take a sample from each tube at the beginning and at the end of experiment. Not all the content of the tube A or the tube B undergo Fehling test or iodine test, but we take a small sample of tube A and of tube B at the beginning of the experiment and at the end of the experiment. The results are represented here. Okay, for iodine test, at the beginning of experiment, we take a sample of tube A and one of tube B, and we make iodine test. We apply iodine test for tubes A and B. The color will be dark blue, indicating what? Indicating the presence of starch. So starch present in tube A and in tube B at the beginning of experiment. In the other hand, we take another sample of tube A and B, of tubes A and B, and we apply the Fehling solution, Fehling test. After heating, the color of A is blue, and the same of B, the color is blue, indicating the absence of reducing sugar. This is at the beginning of experiment. At the end of the experiment, the same iodine test is applied on tubes A and B after taking a small sample of each one of them. Starch disappears in tube A at the end of experiment. At the beginning, there is starch in A. At the end, starch disappears. Look at Fehling solution, Fehling test. At the end, at the beginning of the experiment, it gives negative result for Fehling test, which indicates that there is no reducing sugar in tube A. At the end of the experiment, a reducing sugar appears. Look at the results. Starch disappears at the end of experiment in tube A, and reducing sugar appears at the end of experiment in tube A. Look at tube B, where starch is put with water. Nothing changed, okay? There is starch at the beginning and at the end. There is no sugar at the beginning and no sugar at the end. Okay, what does it mean? Look at the questions by referring to the experiment. Indicate the variable factor. What is the variable factor here? In order to know the variable factor, we make a little comparison of the conditions of the experiment between A and B, starch with saliva, starch with water. So the variable factor is the presence or the absence of saliva, okay? Because water exists in both tubes. So the presence of saliva is the variable factor. State the role of tube B. Okay, tube B, nothing changed in tube B. We put starch with water in order to compare the results between A and B. Look at the results here. The result that appears in A and that in B. So in order to compare the results after put some conditions, we use tube B, which is the control tube. Number 3, describe the experiment. To describe the experiment, we should use some keywords. We take two test tubes, A and B. We put in each one of them an amount of starch. Then we add starch to tube A, and we add water to tube B. Finally, we place the two tubes A and B in water bath at 37 degrees for 20 minutes. So in order to describe an experiment, we should use take, put, add, place as keywords in order to clarify the steps of the experiment. Interpret the results of experiment. Interpret means make an analyze, then the significance. In tube A, iodine test gives dark blue color in tube A at the beginning of experiment. This indicates the presence of starch in tube A at the beginning of experiment. At the end of experiment, it gives dark, it gives brown orange. This indicates the absence of starch in the tube A at the end of experiment. For tube B, iodine test gives dark blue color at the beginning and at the end of experiment, indicating the presence of starch in tube B at the beginning and at the end of experiment. For Fehling test, tubes A and B undergo Fehling test. At the beginning of experiment, the coloration was blue, indicating the absence of reducing sugar. At the end of experiment, tube A gives brick red precipitate, and tube B, the coloration will be blue. This indicates that reducing sugar appears only in tube A. Give the corresponding conclusion. The conclusion was the starch, which is called also amylose, is digested by salivary amylase, the amylase, which is the enzyme present in the saliva, and simplified into molecules of maltose. Okay, so starch molecules, complex molecule of starch, is digested by salivary amylase present in saliva into maltose. This is the end of our lesson. We should remind about the hydrolysis. The hydrolysis, which is the decomposition of complex molecules in the presence of water, and digestion that occurs by the help of enzyme, um, uses enzyme, which is a chemical substance secreted by the body and ensures the simplification of complex molecules. This is that we call molecular simplification. Thank you for watching, and have a nice time.